Greetings. Today we're going to be reading from Creating Mind, How the Brain Works by John E. Dowling. Today's recording is from pages 41 through 59. We'll be reading from chapter 3, Drugs and the Brain. Tess was the eldest of 10 children, born to a passive mother and an alcoholic father, in the poorest public housing project in our city. She was abused in childhood in the concrete physical and sexual senses which everybody understands as abuse. When Tess was 12, her father died, and her mother entered a clinical depression from which she never recovered. Tess, one of those inexplicably resilient children who flourish without any apparent source of sustenance, took over the family. She managed to remain in school herself and in time to steer all 9 siblings into stable jobs and marriages. Her own marriage was less successful. At 17, she married an older man, in part to provide a base outside the project for her younger brothers and sisters, whom she immediately took in. She never went to the movies alone with her husband. The children came along. The weight of the family was always on her shoulders. Tess' husband was alcoholic and abusive when drunk. Tess struggled to help him stop drinking, but to no avail. The marriage soon became loveless. It collapsed once the children, Tess' siblings, were grown and one of the marriage's central purposes had disappeared. Meanwhile, Tess had made a business career out of her skills at driving, inspiring, and nurturing others. She achieved a reputation as an administrator capable of turning around struggling companies by addressing issues of organization and employee morale, and she rose to a high level in a large corporation. That her personal life was unhappy should not have been surprising. Tess stumbled from one prolonged affair with an abusive married man to another. As these degrading relationships ended, she would suffer severe demoralization. The current episode had lasted months, and despite a psychotherapy in which Tess willingly faced the difficult aspect of her life, she was now becoming progressively less energetic and more unhappy. It was this condition I hoped to treat in order to spare Tess the chronic and unremitting depression that had taken hold in her mother when she was Tess's age. Though I had learned some of the story before my consultation with Tess, the woman, when I met her, surprised me. She was utterly charming. She was pleasure to be with, even depressed. I ran down the list of signs and symptoms, and she had them all. Tears and sadness, absence of hope, inability to experience pleasure, feelings of worthlessness, loss of sleep and appetite, guilty ruminations, poor memory, and concentration. 2 weeks after starting Prozac, Tess appeared at the office to say she was no longer feeling weary. In retrospect, she said, she had been depleted of energy for as long as she could remember, had almost not known what it was to feel rested and hopeful. She had been depressed. It now seemed to her her whole life. She was astonished at the sensation of being free of depression. She looked different, and once more relaxed and energetic, more available than I had seen her, as if the person hinted in her eyes had taken over. She laughed more frequently, and the quality of her laughter was different, no longer measured but lively, even teasing. With this new demeanor came a new social life, one that did not unfold slowly as a result of a struggle to integrate disparate parts of the self, but seemed rather to appear instantly and full-blown. Excerpted from Peter D. Kramer, Listening to Prozac. At synapses, neurons can only be excited or inhibited. They can also be modified or modulated in a variety of ways. The second type of synaptic action is called neuromodulation. I have already described how neurons are excited or inhibited at synapses. Now, I'll detail how neurons are modulated by synaptic action. Why are these neuromodulatory processes important? Because long-term changes that occur in the brain and resulting phenomena such as remembering and learning are thought to happen because of neuromodulatory synaptic action. But also, many chemicals that alter mental states, such as LSD and the amphetamines, and diseases that cause thought disorders, such as schizophrenia, can be related to alterations in neuromodulatory synaptic transmission. And as the story of Tess described above illustrates, inducing alterations in levels of neuromodulatory substances in the brain by drugs such as Prozac can provide striking relief from serious psychiatric disorders such as deep depression. We know that transmission at most brain synapses is chemical. A substance released from one neuron diffuses to an adjacent neuron and induces changes in the neuron. A few synapses are electrical. As such synapses, the cell bodies of two adjacent neurons become closely opposed, and protein channels create a bridge between the contracting cells. These channels enable ions to flow directly from one cell to another. A changing membrane voltage in one cell results in a rapid changing voltage in the contacted cell as the ions flow from one cell to the next. Such transmission is very fast and can go in either direction. Electrical synapses then synchronize activity between neurons or permit reciprocal interactions between neurons. But signals cannot be amplified at electrical synapses. Amplification is possible only at chemical synapses. Also, signal polarity cannot be changed at electrical synapses, from excitation in one cell, for example, to inhibition in the adjacent cell, or vice versa. Not only that, there is little in the way of signal modification at electrical synapses. Those chemical synapses are predominant in the brain because they allow for a richness of interaction between neurons. Neurotransmitters and neuromodulators. As many as 50 different substances may be released at chemical synapses in the brain. Usually, no more than one or two substances are released at any one synapse, and it is generally believed that all the synapses made by a neuron release the same substance or substances. So neurons have a lot of diversity and specificity in relation to the substances they release at their synapses. It is convenient to classify the substances released at synaptic sites into two categories, neurotransmitters and neuromodulators, based on their mode of action on postsynaptic cells. Neurotransmitters are substances that interact directly with channels in the postsynaptic membrane. They mediate fast excitatory or inhibitory synaptic action. When a neurotransmitter activates such channels, they open and allow certain ions to cross the membrane. As you already know, when positively charged ions such as sodium cross the membrane, the inside of the cell becomes more positive and the neuron is more likely to generate an action potential. It is excited. We'll make a little parenthesis here since we have not talked about action potentials in class. Essentially, think about action potentials as electricity from one cell to the next. So as of now, what you should think about is that cells can communicate with each other with chemicals, and these chemicals produce electricity from one cell to the next. Going back to the text. Conversely, if negatively charged ions such as chloride cross the membrane, the inside of the cell becomes more negative and the cell is less likely to generate an action potential. It is inhibited. We'll make a parenthesis here for our reading. So it is possible to make cells less excitable, meaning less responsive, and that is called inhibition. Excitation is when the cells are more excitable. We'll go back to the text. These excitatory and inhibitory interactions are quite fast. Once the synapse is activated, it takes less than half a millisecond, 0.0005 of a second, for a voltage change to occur in the postsynaptic cell. Furthermore, the changing voltage in the postsynaptic cell lasts only a short time, between 10 and 100 milliseconds, or no more than zero point 0.1 of a second. We'll make a parenthesis in the text here again. As you think about the magnitude of changes that take place between neurons, think about a battery. So batteries have different types of voltages, and so do cells. But the voltage in a battery could be 3, 6, maybe 9, or even 12 volts. But cells could also have voltages, and these voltages are very, very small amounts. And so we want to think about voltage as the ability to make a change that is going to result in work. And this work is a result of electricity, which in turn is the result of a chemical, a neurotransmitter, such as, for example, epinephrine, which, as we know in class, can also be called adrenaline. Now let's go back to the text and let's think about neuromodulators. So neuromodulators act quite differently on neurons. When neuromodulators are released as synapses, they diffuse to the postsynaptic membrane, where they interact with membrane proteins called receptors, which are linked to intracellular enzyme systems. Activating these receptors does not directly open the channels in the membrane. Rather, the neuromodulator's action is mediated by biochemical changes in the postsynaptic neuron. In a typical situation, intracellular enzymes are activated and synthesize small second messenger molecules. The first messenger is the modulator. Many aspects of neural cell function can be altered, from the properties of the channel proteins in the membrane to the expression of genes in the nucleus. In this matter, profound physiological and structural changes can happen in neurons as a result of neuromodulation. Neuromodulatory effects typically have a slow onset, usually seconds, and then can last for long periods of time, minutes, hours, days, or even longer. As noted above, the evidence is accumulating that long-term changes in the brain, which underlie phenomena such as memory and learning, result from neuromodulatory synaptic interactions. Some substances released as synapses act exclusively as neurotransmitters. Others are solely neuromodulators. Most substances released at synapses can act as both. At some synapses, they interact with membrane channels postsynaptically, while at other synapses, they interact with membrane receptors linked to intracellular enzyme systems. The neurotransmitter acetylcholine is a typical example. It is the neurotransmitter used at all synapses between nerve and muscle invertebrates, the neuromuscular junction, but it can also activate membrane receptors in the brain. Linked to biochemical systems. These two kinds of actions can be distinguished by pharmacological means. Certain drugs will block one action but not the other, and vice versa. Curare, we'll make a parenthesis from the text. Curare is a substance which actually is a toxin that is naturally produced by some plants. We'll go back to the text. Curare blocks the action of acetylcholine at the neuromuscular junction, whereas the drug atropine blocks the effect of acetylcholine on membrane receptors. We'll make a parenthesis from the text. Curare, what it does is that it essentially paralyzes the individual by blocking the ability of the brain to communicate with muscles. We'll go back to the text. Atropine has no effect on the neuromuscular junction, and curare does not block the neuromodulatory action of acetylcholine on membrane receptors. Some agents can specifically activate one or the other action of acetylcholine. Nicotine activates a membrane channel specific to acetylcholine, while muscarine activates acetylcholine receptors linked to enzyme systems. These two effects are often referred to as nicotinic or muscarinic actions of acetylcholine. So we'll make a parenthesis from the text. So far, we're thinking about different types of chemicals that have different types of actions on the neurons. And so we want to think about the actions of nicotine, which we'll review in lecture. We also want to think about the actions of curare, which is a toxin that blocks the ability of the brain to communicate with the muscles. And we'll also think about a compound, which also is a chemical atropine that then can return the or release the blockage from curare onto the muscles. We'll go back to the text. Figure 16 shows a neuromodulatory system works. The system chosen for illustration uses an enzyme, adenylate cyclase, that synthesizes a second messenger molecule called cyclic AMP. A neuromodulator, the first messenger, released from vesicles in the presynaptic terminal, interacts with the receptors on the postsynaptic membrane, and it thereby activates them. The activator receptors now interact with an intermediate protein called a G protein, which in turn interacts with adenyl cyclase and activates it. We'll make a parenthesis in the text. So essentially what this means is that a chemical that we call a neurotransmitter is released from one cell, and the second cell that receives that neurotransmitter then releases within its cell a molecule that is called a second messenger. Go back to the text. The Nobel Prize for Physiology and Medicine was given to Alfred Gilman and Martin Rodbell in 1994 for the discovery of G proteins. Virtually all known membrane receptors linked to intracellular enzyme systems include the light-sensitive molecules in photoreceptor cells, the odor-sensitive proteins in olfactory cells, and the neuromodulatory receptors on neurons. Interact first with G proteins. These are a number of G proteins known that can activate or inhibit a variety of intracellular enzymes. So we'll make a parenthesis from the text. Essentially what this means is that we're able to see, to smell, and to hear because of these different types of proteins. We'll go back to proteins called G proteins. We'll go back to the text. Regarding the system in Figure 16, activated adenyl cyclase promotes the conversion of the molecule adenosine triphosphate, ATP, which is ubiquitous. Ubiquitous means that it's present everywhere. So, which is ubiquitous in cells and stores and provides energy into a smaller molecule called cyclic AMP. Cyclic AMP exerts its effect by activating another enzyme termed a kinase that adds a phosphate group to cellular constituents, usually proteins. This process, called phosphorylation, is a favorite way for cells to activate or inactivate biochemical reactions or to modify proteins' properties. The kinase activated by cyclic AMP is called a protein kinase A, or pKa. Let's take a look at Figure 16. As we take a look at Figure 16, you'll notice that the presynaptic terminal, all this means is the cell that releases the molecule that we call a neurotransmitter. The dots between the cells represent the compound or the chemical that we call a neurotransmitter. Some examples of neurotransmitters that we have thought about are acetylcholine, epinephrine, norepinephrine. And so the cell that releases the neurotransmitter onto a cell that receives the neurotransmitter. That neurotransmitter is then bound to a receptor, which then activates a G protein. So for us, what we want to think about or we want to concentrate on is the release of a neurotransmitter, which then binds to a neurotransmitter receptor, which in turn binds to a G protein, which then produces cyclic AMP. Lowercase c is for cyclic AMP. And cyclic AMP, as you'll notice, comes from ATP, adenosine triphosphate, which is what provides energy for the cell. Let's go back to the text, bottom of page 46. So it can cause effects at many levels of the cell, including the nucleus, the cytoplasm, and the membrane. In the nucleus, genes can be turned on or off. In the cytoplasm, enzymes can be activated or inactivated, including those involved in protein synthesis. And at the membrane, ion channels and other membrane proteins can be altered. For example, phosphorylation of a channel. Phosphorylation, all it means is a chemical type of reaction that adds a phosphate to another molecule. The significance of phosphorylation in cells is that adding a phosphate to another molecule usually activates that particular molecule. We'll go back to the text. Phosphorylation of a channel can modify its sensitivity to a neurotransmitter, or how long the channel remains open after its activation by a neurotransmitter, or even its ionic specificity, that is, which ions it will admit into the cell. Another interesting feature of neuromodulation is that a substance released at a synapse can affect the terminal releasing it. Those synaptic terminals can have their own receptors, termed autoreceptors, that respond to the neuromodulator. In our example in figure 16, autoreceptors are linked to the cyclic AMP cascade. The activated kinase may, for example, phosphorylate enzymes that synthesize the substance released by the terminal. With such mechanisms, a neuromodulator can regulate how much substance is made by the terminal. That is, a terminal can modify its own properties by feedback mechanisms. What this last paragraph means is that chemicals can modulate or regulate how much of the neurotransmitter is released. Back to the text. Even though several second messenger cascades are known, the list is far from complete. Cyclic AMP is the most extensive studied second messenger and the one best known. Other second messengers activate other kinases, and any kinase is very specific with regard to the molecules it can phosphorylate. One neuromodulator at synapse can activate more than one second messenger pathway by way of one receptor, but several G proteins. Thus, a variety of processes can be altered in a cell due to a single neuromodulatory input. Any neuron probably receives many such inputs, so the possibilities for its mode of modulation are considerable. The distinction between synaptic neurotransmission and neuromodulation is sometimes blurred. Some ion channels in the membrane activated by neurotransmitters can admit calcium into neurons, and the admitted calcium can act as a second messenger. The way this works is that the calcium binds to a protein within the cell, termed calmodulin, and the calcium-calmodulin complex interacts with specific kinases termed CAM kinases. So we'll make a parenthesis from the text. Think of calmodulin really as a sponge within the cell that can bind calcium. And when the cell needs a little bit of calcium, the sponge can release the calcium. So think of calmodulin as a sponge for calcium that can release it when needed or can store it when it's not needed. Back to the text. These kinases act like other second messenger activated kinases, phosphorylating cellular constituents and thus activating or inactivating biochemical mechanisms. Long-term changes in vertebrate neurons that relate to memory and learning appear to be mediated by such mechanisms. So as you're reading this text, you are actually making new memories, and you're making new memories because your neurons are putting together new synapses. Remember from the beginning of the classes, we were thinking that our brain cannot make any new neurons, but we're constantly making new synapses, meaning new connections between neurons. And these connections are made by strengthening the neurotransmitter release, and how this neurotransmitter release is going to increase the amount of enzymes that we find in the cell that receives a neurotransmitter. Let's go back to the text on page 48. Classifying synaptic substances. Although as many as 50 substances may be released as synaptic synapses in the human brain, they fall into one of four chemical classes. Two of these classes act mainly as neurotransmitters, acetylcholine and amino acids, and the other two act mainly as neuromodulators, monoamines and neuropeptides. I'll start with the neurotransmitters. We'll make a parenthesis from the text. The ones that we want to be familiar with are the neurotransmitters acetylcholine, and that is the first one that is discussed in the second paragraph. Back to the text. Acetylcholine is the only naturally occurring substance in its class, but other chemicals mimic acetylcholine activity. They will activate acetylcholine channels or receptors. Acetylcholine is usually an excitatory neurotransmitter, but it can exert inhibitory effects through neuromodulatory actions. Indeed, its discovery in the early 1920s by Otto Loewi, a German neurochemist, came about in relation to a slowed heart rate. So acetylcholine is released in the heart when the vagus nerve is cranial nerve number 10. So when the vagus nerve, which regulates the heart, is active, acetylcholine interacts with a receptor on the heart cells linked to a G protein. These G proteins inhibit adenylate cyclase and thus the synthesis of cyclic AMP. The falling cyclic AMP levels diminishes the phosphorylation of certain channels in heart cell membranes, thereby slowing down the heart. What Loewi did was to estimate the vagus nerve in What Loewi did was to stimulate the vagus nerve in an animal and collect the fluid around its heart. He demonstrated that this fluid slowed the heart of another animal whose vagus nerve was not stimulated. It was learned later that the substance released by the vagus nerve is acetylcholine. So from these two chapters, what we learned is that the neurotransmitter acetylcholine, when it is released into the heart, it slows down heart rate. This is normally what happens to all of us when we are at rest. Our nervous system, through cranial nerve number 10, also called the vagus or the vagabond, releases the neurotransmitter acetylcholine to slow down our heart rate. Back to the text. Like acetylcholine, the amino acids released as synapses act mainly as neurotransmitters, although some can interact with membrane receptors linked to second messenger cascades. One amino acid, glutamate, serves as the major excitatory neurotransmitter in the brain. We'll make a parenthesis here. We definitely want to know glutamate because glutamate is a very important neurotransmitter in the brain. Notice that glutamate is excitatory. Back to the text. Two other amino acids, glycine and gamma aminobutyric acid, abbreviated as GABA, GABA, are the major inhibitory neurotransmitters in the brain. So these two are also important to know. Glycine and GABA are inhibitory. Back to the text. Other amino acids such as aspartate, cysteine, and histamine have excitatory or inhibitory actions on neurons, but not much is yet known about their roles in synaptic transmission. So to recap this section, we want to make sure that we know acetylcholine. We also want to make sure that we know glutamate, which is excitatory on the brain. We want to make sure that we know glycine and GABA, which are inhibitory to the brain. Back to the text, glutamate, GABA, and glycine are closely related structurally, as you can see on figure 17. The molecule glutamate becomes GABA when a carbon atom and two oxygen atoms are removed. Without its tail, the molecule is glycine. Let's take a look at that figure. The top of figure 17 shows the structure of glutamate. The second one is GABA. You'll notice that GABA has this group that has been removed. The point for our class is not to take a look or memorize the chemical structure, it's just to realize that glutamate, which is an excitatory neurotransmitter, is very closely related to GABA, which is an inhibitory neurotransmitter. And then GABA, you'll notice, is larger, as well as glutamate, as compared to glycine. Glycine is a very small neurotransmitter, and glycine happens to also be an amino acid. Let's go back to the text. Glutamate activates two types of channels. One allows sodium to enter a cell, thus exciting it. The other channel allows both sodium and calcium to enter the cell, which excites the neurons and also activates calmodulin. Remember that calmodulin is a sponge that takes up calcium and can also release it. Back to the text. Activates calmodulin and CAM kinases. Thus, when a neuron possesses this channel, it can be both excited and modulated at the same synapse. We have evidence that this channel plays a role in storing memories. When we experience something, not only are our neurons excited, but they must also be modified in some way to account for memory. How this happens is not well understood, but neuromodulatory pathways activated at these glutamate synapses may underlie such long-term changes in neurons. GABA and glycine mainly activate channels that allow chloride to flow into neurons. This results in inhibition of the neurons. The GABA-activated chlorine channel is of particular interest because its properties are specifically altered by these kinds of drugs: barbiturates, benzodiazepines, and alcohol. Barbiturates and benzodiazepines are used to treat anxiety, and at low concentrations, alcohol can also relieve anxiety and promote relaxation. The benzodiazepines were introduced in the 1960s and the early 1980s. They were used by enormous numbers of people. A variety of benzodiazepines are available. The best known is diazepam or Valium. By the early 1980s, about 20% of the women and 10% of the men in England were taking a benzodiazepine at one time or another during the year. Barbiturates, benzodiazepines, and alcohol all potentiate the GABA-activated chloride channels. Potentiate means to increase the response. When these drugs are present, the channels allow more chloride to cross the membrane. Hence, the postsynaptic neurons are more strongly inhibited. How increased inhibition relieves anxiety is not understood. What is known is that the barbiturates and benzodiazepines interact at specific sites on the GABA channel, and the sites are different from the site where GABA interacts with the molecule. The fact that these exist sites specific for a drug on a membrane channel or receptor suggests that an endogenous, remember endogenous means produced by the body, that an endogenous brain substance naturally interacts with the site. As yet, no endogenous benzodiazepines or barbiturate-like substances have been identified. Monoamines, of which there are two types, catecholamines and indolamines, serve almost exclusively as neuromodulators. Catecholamines are derived from the amino acid tyrosine, and three catecholamines, dopamine, neurotransmitter, and epinephrine, are important in brain function. Indolamines are derived from the amino acid tryptophan, and one indolamine, serotonin, is a key substance released at brain synapses. Figure 18 presents the structure of dopamine and serotonin and the amino acids from which derive. So from this chapter, what we want to know is the neurotransmitters, dopamine, norepinephrine, and epinephrine. We're also going to see that they derive from an amino acid, means one component of proteins from our diet. Monoamines are critical in regulation in the brain's mood. It's effective It's affective and arousal states. Drugs that alter transmission at synapses using one of the monoamines or that alter the levels of these substances at synaptic sites often dramatically change a person's mood or other mental state. The hallucinogen lysergic acid diethylamine, or LSD, is a good example because it interacts with membrane receptors activated by serotonin. We'll make a parenthesis in the text. Serotonin is an extremely important neurotransmitter to know, since we're going to learn that serotonin is one of the very important neurotransmitters that regulate mood. The more serotonin available, the happier or more elated that we are. The less serotonin available, the sadder or more clinically depressed we may be. Back to the text, thereby preventing serotonin from interacting with the receptors. The amphetamines, another example, are brain stimulants that induce hyperactivity and inability to sleep. They increase levels of catecholamines at synapses, particularly dopamine, by releasing the substances from nerve terminals and by interfering with the reuptake of catecholamines into the presynaptic terminals. From here, what we want to know is the relationship of tyrosine, which is an amino acid, meaning a component of proteins, which we take in our diet, how tyrosine is the molecule that is used to make dopamine. Another amino acid, which is a component of proteins that we take from our diet, is tryptophan. And tryptophan is what is used to make serotonin. So for our class, we're not going to worry about the chemical structure. What we want to just take a look at is that dopamine comes from a amino acid called tyrosine, and serotonin comes from an amino acid called tryptophan. Let's go to the bottom of page 51. Dopamine, Parkinson's disease, and schizophrenia. The monoamine's range of activities is broad and by no means entirely known. For example, two quite different diseases of the brain, Parkinson's disease and schizophrenia, have been related to altered dopamine levels. Parkinson's disease is primarily a disease of the motor system. Patients suffering from Parkinson's disease develop a severe tremor and have trouble initiating movements. They also become rigid and their movements are characteristically slow. In the late 1950s, it was discovered that the brain's dopamine content was low in patients suffering from Parkinson's disease. Much of the brain's dopamine is in its nuclei, termed the basal ganglia, that are involved in the initiation of movement. In one basal ganglia nucleus, more than 90% of the dopamine may be lost in the disease. In the early 1960s, a drug therapy for Parkinson's disease was introduced that raised the brain's dopamine levels. The drug L-DOPA, a precursor of dopamine, is effective because it passes readily from the blood to the brain. Dopamine itself does not do that. It is blocked by the blood-brain barrier, which excludes many substances, good and bad, from entering the brain. Once in the brain, L-DOPA is converted to dopamine and increases the brain's level of the drug. Although L-DOPA causes side effects and sometimes induces schizophrenia-like symptoms, it is successful in many patients and significantly reduces symptoms of Parkinson's disease. So essentially from this paragraph, what we want to think about is that normally we produce a compound or a neurotransmitter called dopamine. That is what is made in our brain. Now, if dopamine is not produced by our brain pharmacologically, another compound called L-DOPA can be given to individuals. The reason that individuals are giving L-DOPA and not dopamine is because L-DOPA can go into the brain while dopamine cannot go into the brain. Remember that dopamine is made within the brain. Let's go back to the text. In recent years, another therapy for Parkinson's disease has been tried with mixed results. With this therapy, cells are transplanted into the brain to release dopamine. Such transplants are expected to raise dopamine levels. Though neural and non-neural cells have been tried, the best results so far have been with fetal cells derived from basal ganglia nuclei. Although results are promising, cell transplantation is not yet as effective drug therapy. Schizophrenia is a severe mental disorder in which patients' judgment is impaired and they lose contact with reality. Schizophrenic symptoms include thought disorders in which thoughts and speech are disconnected, delusions and hallucinations, mood disorders including depression, anxiety, and euphoria. Euphoria means extreme abnormal happiness, restlessness or inactivity, and withdrawal from social interactions. Two observations suggest that schizophrenia may be due to alterations in dopamine synaptic transmission. First, the amphetamines. Amphetamines is a compound that is not made by the body, is a chemical that can be given to some individuals clinically, but mostly is taken as a drug. Amphetamines, what they do is that they excite the brain. First, the amphetamines that raise brain dopamine levels can induce in humans a state that closely resembles schizophrenia. Second, the standard treatment for schizophrenia is drugs that block dopamine receptors. These drugs prevent dopamine from activating dopamine receptors. The most commonly prescribed agents are haloperidol and clozapine, and they are quite effective in treating many symptoms, especially delusions and hallucinations. For many schizophrenics, these antipsychotic drugs have been a godsend because they enable them to function reasonably well in society. So, two important antipsychotic drugs to know that are still used today are haloperidol and clozapine. So, haloperidol and clozapine are antipsychotic drugs that are prescribed to individuals that are schizophrenic or that they may have hallucinations. Although indirect evidence suggests that the underlying cause of schizophrenia involves dopamine synapses, we really have no direct evidence. As yet, no study has provided unequivocal evidence for altered dopamine neurotransmission in the brains of patients suffering from schizophrenia. No region of the brain has been found, for example, that possesses excess levels of dopamine. A problem with treating any brain disease with drugs is side effects. Schizophrenics are given drugs that block dopamine action, thus the presumption is that their brains contain excess dopamine. Parkinson's disease, in contrast, is due to a deficit of dopamine. Do schizophrenics treated with dopamine blocking agents develop Parkinson's like symptoms? They often do, which limits the amounts and types of dopamine blocking agents that can be given to schizophrenics. Conversely, some people with Parkinson's disease develop schizophrenic-like symptoms given with L-DOPA, presumably because dopamine levels are increased in some regions of the brain. For certain patients with schizophrenic-like symptoms are so severe that L-DOPA therapy must be discontinued. Withdrawing the L-DOPA therapy reduces the schizophrenia-like symptoms, but the Parkinsonian symptoms reappear. So from the paragraphs that we have read so far, one of the things that we have learned is that dopamine levels in different parts of the brain can cause different types of conditions if they are too high or too low. For example, Parkinson's disease can be caused or is caused if dopamine levels are too low in a particular portion of the brain. And schizophrenia is caused by dopamine levels being too high in other